Welcome. I'm Ashley Best. I am from the University of Georgia, and I'm the Newton County Extension Agent. Thank you for joining the Organic Conference today. I will be the moderator for this session. I'd like to thank Everlizer for being the sponsor for the 2025 Organic Educational Sessions. So at this time, I would like to welcome Dr. Laurel Dunn. She is with the University of Georgia.
All right, thank you for the introduction. We'll explain what we mean by BSOWs in a minute, but we're going to spend some time discussing some of our food safety concerns, which I really hate the word concerns. I guess they're with poultry litter and our litter-based products. I would like to maybe more say just like some considerations to have if you are using poultry litter or some of these products, since we do rely heavily on them in organic systems, and they are a great fertilizer source. So as a food safety person, I think people expect me to come up here and totally hate on some of our biological soil amendments, which is where the BSOW came from, but they definitely do have an important place in organic production, and we just need to make sure that we're using them correctly. So, are we familiar with the produce safety rule in this room? Anyone? Anyone? All right, I'm seeing some heads, so, and a trainer waving, so, all right. So for those of you that maybe aren't familiar, or for those of you that are familiar but might need a little refresher, produce safety rule is relatively new on the regulatory framework. From the standpoint of produce farms weren't ever really regulated from a food safety standpoint at a national or federal level until the 2000s, or really 2011. So the long name for it is the Standards for Growing, Harvesting, Packing, and Holding of Produce for Human Consumption. So if you do any of those activities, you might fall under the rule. And it really is a tiered program where it's really addressing most of our farms that are making more than about $30,000 a year. So the magic number is $25,000. That number is adjusted for inflation on 2011 dollars. So it ends up being farms that are making about $32,000, $33,000 a year. A lot of times have to follow this federal regulation called the Produce Safety Rule. In the state of Georgia, it is administered by the Georgia State Department of Agriculture through the FDA. In most of our southern region states, whatever state you're in, your Department of Agriculture will be the one actually conducting the inspections on your farm. So BSOW is kind of the cute acronym that came out of the produce safety rule, or at least as what the food safety folks at University of Florida are calling it. So we have this long term with our biological soil amendments of animal origin. Simply put, animal poop is really the most frequent BSOW that we come across, but we can have a lot of compounds. If it just comes from an animal, it's probably this biological soil amendment of animal origin that you are adding to the soil for fertility, for conditioning, for other purposes. So it often ends up being some of our waste from animal agriculture. A lot of times we'll have some commercial formulations of things, and we'll get into some of those and some of the ones that we have looked at Georgia and at UF. Obviously, if you're in an area like the Southeast where we have a lot of poultry production, we tend to go really in the direction of poultry litter or some of these poultry-based products. If you're in an area that's heavy swine production, you might be using pig feces or dairy manure or whatever the frequent animal agriculture that you encounter tends to be. We use it a lot because it is one of our best ways to meet some of our organic requirements, so you're not adding those synthetic nutrient sources. So again, definitely has its place in the produce safety rule. Produce safety rule and the Food and Drug Administration do allow the use of these, but we do have just some things to keep in mind if you're going to be using them safely to make sure that you're not going to be running into any issues from a food safety standpoint. So our FDA definition, I promise I think this is the only definition I have for you, but when FDA is describing a biological soil amendment of animal origin, they're saying something that consists in whole or part of materials of animal origin, such as manure, it's going to be our most frequent one, but also some of our non-fecal byproducts, including animal mortalities, table waste, feather meal, bone meal, blood meal, fish emulsion. All of those are going to fall under this BSOW definition. So we have a lot of things included in that. Table waste is one, particularly I think some of our smaller organic farms that might be getting waste from restaurants and things like that. If there's the potential for humans to have been served that food, there's a potential for contamination. So if you're getting things from local restaurants to apply as a fertilizer source, FDA would consider that a biological soil amendment of animal origin. So we have kind of two different categories of these. Our treated ones are the ones that our food microbiologists love. So things like compost, things like bone meal, where you're getting a heat process on that material. We're mostly killing a good portion, or at least most of the foodborne pathogens on those. So these are going to be things like our fish emulsion, bone or feather meal, compost, whether you make that on your own farm or if you're buying that from someone else. Usually the compost process, just by how you make it, is letting that pile heat up naturally through those microbial processes. And so you're getting enough heat to kill foodborne pathogens. The nice thing about food safety, a lot of times it goes on with plant pathogen control. So if you're composting and killing foodborne pathogens, you're also probably killing a lot of your plant pathogens and weeds and things like that. So a lot of our things in food safety can actually benefit some of your production needs as well. Because we are treating these or heating these, we have a pretty low risk to foodborne contamination. We always say in food safety your risk is low because we do have products that are historically very safe and something goes wrong and we have an accident with them. So we never say there's a no risk food safety product out there, but for these it's typically a pretty low risk because it's gotten that thermal process or that acid process or something that has reduced those foodborne pathogens. So then we have our untreated basows, which again we can use. The FDA allows the usage of these, but we're going to say these are a potentially higher risk. I won't say across the board they're always going to be less safe, but these are going to be ones that are not treated to control microorganisms. So, our poultry litter, if we're pulling it straight out of the house, or even if we're just stacking it for a little bit of time, but we're not actually measuring how hot it gets, FDA does not consider that to be compost. So if you're pulling it out of a dry stack shed, but you've never gotten a temperature on how hot that pile got, they're gonna call that an untreated biological soil amendment. Cow manure, pig slurry, those table scraps, again, are going to be considered in that. And just the concern with this is we know a lot of our foodborne pathogens are zoonotic or they're coming from our animals. So if we tend to think things like E. coli, the food we tend to think of as ground beef. Beef tend to have E. coli. Salmonella tends to be carried by chickens. So we tend to assume there's that association there that if it's in those animal products, it's going to be in their feces. It's going to be in whatever fertilizer we're using if it doesn't get that treatment. So when I was at UF for a little bit, we did a survey, and I just have one of the surveys up here. We're collecting poultry litter out of a lot of dry stack sheds. This tended to be older poultry litter, so our salmonella levels ended up being about 7% in the samples we collected. We collected about 490 samples. 7% of those had salmonella on them. We visited farms multiple times, and so for these farms that got visited, we did find salmonella in 46%, or almost half of the farms that we collected samples from, so at these different times of sampling. So 7% might not be that alarming to you. I should have put up another table that one of my colleagues at UF did a similar study, but they were testing younger, if you will, poultry litter, and they were getting more like 50% positive rates of salmonella in just fresh poultry litter out of the chicken house or recently put in the dry stack shed. So we know it's there, we know it's something to be considered, and we just want to make sure that if we're using these things, we're doing them safely. We obviously like our biological soil amendments because they do have those benefits. That was why FDA didn't come down on them. We have increased nutrient levels usually. Once we start composting, we lose a lot of nitrogen and a lot of other compounds in them. We save a lot of time if we're not composting. Particularly in an area like Georgia or where we have a lot of really concentrated animal production, it's really our best way to reduce some of our waste from animal agriculture is by using it as a nutrient source. So FDA was not going to come down and say, hey, you cannot use this at all, but they do really encourage that you use it safely. And we'll get into what that requirement is from the produce safety rule. So just to show a little bit of work that we did, we conducted a study in cooperation with University of Florida, our food safety colleagues down there. So our UGA team had a plot at the Vidalia station, and our UF team had a plot at their station in Live Oak, Florida. And so we took about 3 by 9 foot plots here, and we amended the soil. Either we did not amend the soil, but we just tilled it. We applied poultry litter, or we applied poultry pellets at the recommended rate for the crop that we were trying to produce at the time. we then inoculated those same plots with a non-pathogenic E. coli. So this is an E. coli that if our neighbors doing other experiments adjacent to us, if some of our E. coli wandered into their plots, they weren't going to get anyone sick, but there was still this E. coli strain that we could track really easily in the soil and pretty confidently get it back and know what our numbers were, so we could track the die-off of that. So our E. coli strain was this E. coli TVS 353, a non-pathogen that we can spray in the field and not make anyone sick. And we actually did the study for 2 years at both locations. We tracked this E. coli in these 3 different types of plots for about a 140-day production cycle. So we were going starting in November through April for these collection dates. And so just what's interesting with these is I have both our Vidalia data from UGA and our live oak data. We start at pretty high levels of E. coli up here, so we have about anywhere from 4 to about 5 log CFU per gram of E. coli is what we initially started with at the beginning of the season that we put into these production plots. And then we just went back repeatedly between Athens and Vidalia. My students got a lot of miles in collecting these samples, and as you can see, we do get a pretty consistent die-off throughout the production season. It looks like around day 70, our poultry litter amended plots, we were below 0 here. We were no longer really able to detect it in those poultry litter plots. Our unamended plots, lose my voice. I've given a lot of talks today. We got pretty low levels here, but we were still detecting a little bit of E. coli. When we got to about day 85 to about day 140, there was no change, but we still detected E. coli every time we went out to the plot in our unamended plots. What I really want to point out here is this red line is our heat-treated poultry pellets. And people tend to really get comfortable with heat-treated poultry pellets because they think that they're a sterile, they're a treated compound that we're adding to these production fields. And what we're really kind of seeing with these, and this trend that we're seeing in other similar studies too, is that there's something going on with these pellets that they tend to really encourage microbial survival over time when you're using them in a production environment. So as you can see, our red line really took a lot longer. Day 60, we still are getting 3 log CFU per gram out of these soil samples, so still a significant amount. And it really wasn't until this period between day 110 and 140 that we got that final good drop-off here, but we're still detecting it at least at higher levels than that poultry litter amended plot. So there's just something interesting going on there. I'm a big fan of using poultry pellets, so I don't want you to take away from this that I'm saying don't use them. Just saying be aware that there's still some microbial concerns when you're applying them. Make sure that you're not touching that portion of the crop that you were planning to harvest, because our soils are still seeming to be impacted by the application of those. And kind of the same thing happened in live oak. So it's always encouraging when you have 2 different institutions doing the exact same study on the exact same days for 2 whole production seasons, and your results match up, because that was a lot of coordination going on. But they really saw the same trend. So their controls dropped, relatively quickly. Their controls dropped a little faster than we did. May have been weather, we're a good distance live oak to Vidalia, so weather may have had an impact on that. But again, with their poultry pellets, they saw a slower decline, and they actually had higher levels out to day 140 of E. coli that, again, it was not coming from the poultry pellets. We artificially added it, but there's something going on there that is really continuing to encourage the growth of these organisms if they're there. So if you had an application where you were applying poultry pellets and you also had contamination from salmonella, or you had wildlife intrusion in your field that you didn't notice, you had salmonella or E. coli or some pathogen deposited in the field, if you're using poultry pellets, you just might have that survival going on. Whether or not that is going to be a public health concern, I don't know that we have enough data on that. So not trying to make anyone nervous about them, just trying to make you aware that that using these, there's still some things going on that we don't fully understand with these. So back to our produce safety rule. the produce safety rule really encourages, if you are going to be using those untreated soil amendments, so I'm not talking the poultry pellets necessarily anymore, now I'm talking you're pulling litter directly out of the house and applying it to your field. We like applying those to some of our lower risk crops. When I say lower risk crop, I'm talking about things we don't eat, so apply it to your cotton, or if you have citrus or something like that, apply it to those things where the crop isn't going to be directly on the ground. Avoid that contact with that edible portion during application. So be really careful when you're land applying things. If you're field spreading, how much dust is being generated? What do you have adjacent to it? Don't be applying things. Don't be applying raw poultry litter on top of things that you're going to be harvesting pretty soon. And then have really good records. So what did you apply? When did you apply it? Where did it come from? If you got it from another farm, where did it come from? If you bought it from a third party, where did you get it from? What's that lot identifying information? In case they did something wrong and misprocessed what they sold you and are recalling that product, you know whether or not the product you used would have been implicated. As far as the produce safety rule, they have a few requirements, and I use the word must strategically here because that is a requirement of the produce safety rule. You must do this if you fall under the produce safety rule. So your biological soil amendments must be handled and stored to prevent contamination, cross-contamination to produce, water, food contact surfaces, etc. So water, don't store your soil amendments right next to your well or right next to your irrigation pond or right next to a production field that you're getting ready to harvest. Make sure that things are stored in a manner where you're not likely to get runoff, wind spread, people tracking it into your packing house, things like that. Another must here, we must not contact produce during application with these biological soil amendments of animal origin, particularly those untreated biological soil amendments of animal origin. And we want to make sure that we're minimizing any contact that we have. As far as an application interval, we'll get to this, I guess, a bit in the next slide. The FDA is not going to tell you how, or has not yet told us, how long you need to wait between when you apply something like poultry litter and when you actually harvest a crop. So right now they're saying they're deferring action until a risk assessment is performed. Some of that data that we showed earlier is the type of data that they're putting in that risk assessment to make a decision. But right now what they're doing is they're directing people to follow the National Organic Program 90 or 120-day application interval. So what this just is if you apply it to a commodity that is unlikely to contact the ground during production, they are going to, I always get my 90 and 120 mixed up, making sure I'm telling you the right thing. So you have 90 days between when you apply that and when you harvest. So you don't want to harvest between that 90 days because we're going to consider that that's directly touching the ground, more likely to pick up contamination if you used a contaminated fertilizer. 120 days is their recommendation for commodities that are likely to contact the ground during production. So, squash, onions, things like that are grown directly in contact with the soil. You want to apply that soil amendment and make sure you have 120 days until you harvest that crop that you're going to be handling. So that is a National Organic Program requirement. So those of you that are organic are doing this anyway. But for the produce safety rule, this is a recommendation. So this is that guidance just because USDA has determined that this is their risk tolerance level. And so FDA is leaning into this until they make their own decision about this. And my guess is they're probably going to land on about the same recommendation. Am I done? I'm getting the hook. So just make sure you're keeping good records. And when in doubt, if you're using untreated biological soil amendments, make sure that you're following that 90 to 120-day interval to reduce some of your food safety risks. Any questions?
Thank you, Dr. Bowden.
All right.
All right, our next speaker is Dr. Tim Coulong. He is from the University of Georgia.
Today I'm going to talk about biochar. I figured this first section after lunch we could talk about soil amendments in organic systems, and so Dr. Dunn was talking about poultry litter and animal byproducts. I wanted to talk a little bit about biochar. I've done some work in biochar. The point of this talk is not to convince anyone to use it or not to use it. Simply, I spent a tremendous amount of time going through the literature, and I wanted to talk to folks about some of the pros and cons and so forth. So of course, biochar, The interest in biochar comes a lot from terra preta, which is a term for dark earth. They are two different things, of course, but for those that are or not familiar, terra preta refers to these regions with very high organic matter. They're found primarily in the Amazon, as well as other areas in the tropics. There's parts of West Africa and so forth where this is found. And digging through a lot of anthropological data, originally these were thought to be due to incompletely combusted biomass. So small areas where there were little pockets of villages or people, communities living. They had fires and so forth. There was uncombusted biomass, and that slowly over millennia made its way into the soil. However, they feel that over time, those individuals saw that there was a benefit to this, and the later distribution pattern showed that it was more of an intentional application. The reason there's an interest in this is because historically Amazon soils are pretty poor. You can see right here, this is a typical soil. And these soils are very old, but the carbon has remained for many, many years and provides a lot of benefits. Those soils tend to be richer, have a lot higher nutrient capacity and so forth. This kind of looks like some of the Piedmont we have in Georgia, so, so hence a lot of the interest. Now, this is not necessarily biochar, but they share a lot of things in common. And so biochar, by definition, is a carbon-rich, highly porous compound produced through pyrolysis. And so the thing about pyrolysis is that you take your feedstock, could be any sort of carbon product, could be wood pellets, could be broccoli leaves. I'll show some data on that in a second. You put it in a reactor in the absence of oxygen or limited oxygen. And so after that process, you get a number of different products. You get syngas, bio-oil, and then biochar, which is a very stable carbon product that can then be put into the environment. And so I'm going to spend a lot of time talking about feedstock and the pyrolysis process today, simply because in the literature, this is what has a huge effect of whether or not your biochar is really doing anything for you. So biochar characteristics, like I said, they vary very widely based on the feedstock, tremendously. And then also pyrolysis conditions, typically temperatures between 330 and 700 C. As your temperature pyrolysis increases, your pH tends to increase, your carbon content increase, surface area increase. Porosity increases up to a point. At some point, the pores become very small micropores, and they're not really that effective in an agricultural environment. And then end content can decrease as well as it burns off. And that's very much affected by what temperature that biochar is produced at. And so I'm going to come back to this many times today, in that the source of your biochar and a lot of the research that's been done, depending upon the pyrolysis conditions, we've seen highly variable results with that. And a lot of it becomes we're not comparing apples to apples, or we're comparing apples to oranges in many cases. So pyrolysis, you can do your own. There's tons of videos out there to do your own pyrolysis at home. I just grabbed this picture from an Oklahoma State publication. On a smaller scale, this is a large barrel here. Here's a 30-gallon barrel within that with the oxygen. It's a low oxygen environment. You put your product in there and heat it up at a certain temperature. It is hard to do high temperature pyrolysis on your own. I've got a paper we cited. I worked with a Borlaug fellow, and it's challenging to do it like this and get it up closer to 600 or 700 degrees. So biochar characteristics, I talked about how important the feedstock is. And so I think if you're using biochar in your farming system, this is actually a publication from Julia Gaskin, who many of you that are longtime organic growers know Julia very, very well. She was a faculty member here at Georgia for many years. And this was something that she did back in 2007. And this was done in Tifton, and they looked at peanut, sawdust, pine chip pellets, pine bark, and hardwood chip. So common Georgia sources of organic matter and carbon. And these, it's hard to see, I realize this now after sitting in the back of the audience, but these are different char temperatures from 370 to 400 to 426. And that actually changed the carbon content as well as the nitrogen content. And so that's a pretty small window there. So you can imagine if you go from 350 to 650, how big of a difference you may actually have. The one thing I did want to point out that you probably can see pretty well in the back is these different sources. Here's the carbon and here's the nitrogen content. And so you go from pine bark-based biochar at about 0.3% in. Of course, peanut straw, peanuts are going to have a lot more nitrogen there, 2%. But depending upon the source of your biochar, if you put this out in your field, you may have very different results. If you have something that's 2% N or 0.3%. Now, a lot of this N is not readily available. A lot of it's kind of sequestered, but there is kind of a short shot of readily available nutrition when you put biochar in. So you will see different characteristics from that. Again, I'm sorry, I thought the screen might be a little bigger, but this is the different pyrolysis temperatures, and then, oh, sorry. I gotta be on the microphone so this is the different pyrolysis temperatures here, and total nitrogen, total carbon. So if you look here, wood bark is 1.8% nitrogen. Just regular hardwood would be 0.3% nitrogen. But even your carbon changes. From wood is 48%, here wood stem 89%. So you can have very big differences in the quality of that biochar just depending upon the source that it comes from. And so I'm going to get to the literature that shows we have a lot of variability in our results, and much of that can be traced back to differences in the source of that. Here you see the carbon to nitrogen ratio. We talked about cover crops this morning. Usually you want, like a 31 ratio. You'll see this one here, this canola straw is 1,600 to 1. That's a lot of carbon, versus some of these are a lot lower. And so I did want to point that out. There are many, many review papers. This will be recorded. I've cited all the papers. There are thousands out there, literally. What do you think we should do in terms of keeping us on time?
It's short. Yes. Yes. I don't want it to be burning you, but yes.
Okay. All right. So a couple other things I wanted to point out in terms of different characteristics of sources of biochar. So this is actually part of a project I worked on a number of years ago. These are dried broccoli leaves that went through pyrolysis at a low temperature. You can see there's not a lot of surface differentiation there. Here's some miscanthus, here's some poplar, here's tomato. The point is that your feedstock is going to have a big effect on the porosity and the chemical qualities of that biochar. A couple things on biochar applications. Typically the application rates are pretty high. I'm going to show some data from a low application rate that we did at planting a number of years ago in a trial. This picture is actually from Dr. Ted McAvoy. He has a graduate student working on this. And so you can see how high the application rates are here in the field and what he's looking at. Most of the studies are in the 10 to 20 ton per acre range. And so when I'm looking at the economics of things, I'm kind of using some of those numbers there. They do range from 2 to 50 tons an acre. Keep in mind, if you do read some of the literature out there, a lot of the pot studies and container studies, they'll put stuff based on percentage. And if you extrapolate that to a per acre rate, in many cases, you're looking at 30 to 50 tons an acre, and that's a lot. You're going to have coverage like that. And so because of that, in terms of cost, it's something you need to think about. NRCS does have a cost share in Florida. I was talking to Dr. McAvoy. I don't believe they have one in Georgia. It's under the Conservation Standard Soil Carbon Amendment practice, where they can do a cost share for biochar. But Ted got me some very recent numbers, and they were about $140 to $176 per ton delivered within a certain radius. So if you're looking at some of the rates in the studies where they're seeing a lot of positive effects, you could be looking at $1,000 an acre and so forth. And so I didn't want to just put that out there. On a small scale, it's a little bit different. Having put out biochar, I also did want to put a note in that the size, the sieve size of the product, since we're talking about application rates, makes a big difference. I did a study for a company where we were actually looking specifically at sieve size, and the really fine biochar, sometimes it's pretty hard to spread. because it goes everywhere. It's very light, highly porous, and so forth. And so we did find that the larger sieve-sized biochar was a lot easier to work with. So a little bit about biochar and yield. I'm going to go through a number of studies where the implications of biochar and yield are varied. Again, it's mostly the biochar source, the rate, crop use, and some other things. This was a radish trial we actually did with biochar several years ago down in Tifton. Okay, I'm going to start motoring through these. In Tifton, and so what we wanted to look at, here's a picture of the field. We actually did biochar at a low rate. And the purpose of this, it was direct seeded, and we set it up so we did a 2x2 offset. So we use this in a lot of row crops in conventional production. We do 2x2 offset with maybe some fertility put out at planting and so forth. So of course you can cut the rate and so on. So we did put that out. This was conventionally managed. It's Tifton soil, so it's very low organic matter. Put it here next to our crop. You can see after seeding here, here's our radishes. That's later on in growth, and then harvest. And so we did hand harvest these. Biochar, like I said, was very tightly banded. We did use low rates, I'll show in a second. We actually had roots growing into the biochar that we had in that 2x2 offset, and you can kind of see them circled there. What we found, and I realize now that further in the back you're not going to be able to see this, but these are, in this column, these are all the different types of biochars and different rates that were applied here, but you'll see this yellow line is our control. So that's nothing put out in that 2x2 offset. And we did have our lowest yield. Now statistically, it wasn't different than these bottom four here, but you can see a number of them up here did out yield that. And you can see our rates were pretty low, 4 pounds per 1,000 feet in that 2x2 offset. So it was a very low application rate, but you see in some cases it was pretty effective on increasing radish yield. The reason we chose radishes is because we wanted to see was there any effect of direct seeding and was there a negative impact on germination. Other trial, there are literally thousands, like thousands and thousands of trials out there. And so, colleagues at the same time, Ajay Nair, he's a colleague of mine at Iowa State University, a very good horticulturist. He did a multi-year study both in potato and pepper. He didn't see a difference. I worked with a gentleman where we did the broccoli-based biochar. We did greenhouse studies. This was in Jordan, actually, as part of his fellowship. We actually found negative effects at very, very high rates. And the reason was because the EC became elevated. And one thing, biochar tends to be more alkaline in terms of pH, and the pH got too high. But again, this was a very high rate. I don't think you could afford to put out 50 tons an acre. This slide here, though, this is a pretty data-dense talk. This actually, I'm going to get feedback from that. I can't stand there, sorry. This slide here was a meta-analysis of a lot of different studies that have been done with biochar. And so what they did was they split up the biochar in terms of high temperature, which is high T, or high carbon, greater than 30%, poor nu, which is low nutrition. They said this was, in the analysis, this was wood and plants, so pine bark, those sort of things. And then rich nu, which is manure and sludges. Now in organics, we're not putting out sludges, and so we're probably not going to encounter a lot of these rich, nutrient-rich new biochars. And then they looked at yield compared to the control. And they found high temperature, high carbon, in general, tended to push yield a little bit more, whereas once you got down to the low temperature pyrolysis, low carbon, they were not seeing any effects positively on yield. And this is several hundred studies that they kind of analyzed. In general, when you looked at nutrient content, the biggest benefits were the rich, new, high carbon. So rich, nutrient, high carbon-based biochars. They saw the biggest benefit in yield. The problem is, again, we're not going to use sludges, and we don't use many animal waste biochars in the United States. And so I did put this in there just to show the impact of both the temperature and your feedstock source. Now, since I'm going to run out of time, sorry, one of the things I wanted to emphasize is that while yield is variable, in general, most of the reviews and the research has shown that there is a very positive effect on soil health attributes in terms of using biochar. And so if you're going to apply, maybe don't look at it as I need to push my yield by X pounds per acre or X bushels per acre, but how am I using this to improve my soil study? And so there was a recent study that Kate actually just sent me the other day out of UC Davis, where they found in processing tomatoes, biochar made from softwood and almond shell, no impact on yield in tomatoes, but there was a positive impact on PHEC, CEC, and all those other soil attributes in their system. And as a soil amendment, of course, it significantly increases cation exchange capacity, provides a lot of places for microbes to live and so forth. We'll go through that. In generally, biochar will decrease the bulk density of soil. Julia Gaskin showed that. I'll show this in a second, by increasing aggregate stability and so forth. And so, again, while you may not see those immediate yield effects, it does go towards, building a healthier soil. It'll actually help increase soil moisture holding capacity. This is done in Tifton. Julia did this a number of years ago. This blue line here, the aqua line, is the control. You see a slight increase in water holding content in that Tifton soil. In Tifton, this would probably be good because we have sandy soils. In Athens, that may not be the most desirable things because our soils tend to hold a lot of water. And then a couple things in terms of microbial activity. Often they'll have a quick release, a quick increase in microbial activity in the soil right after addition because there's some labile carbon and so forth from lower temperature pyrolysis. But in all the studies out there, pyrolysis temperature was the most significant thing impacting microbial activity. So it's not all the same. If you throw out biochar, depending upon the pyrolysis temperature, you may not see a big boost. Okay. Some potential negatives. Some of the feedstocks can contain heavy metals, and when you concentrate those feedstocks, it concentrates those heavy metals in the soil. And so certainly if you're purchasing biochar, ask for an analysis. Most companies, particularly if you're an organic grower, are going to provide that. And it can also contain hydrocarbon contaminants, and that's affected also by the pyrolysis temperature. But there are some potential negatives. So basically, to conclude, the data on yield impact is mixed. Again, feel free to, I put all the citations on this talk, and the talk will be available on the website. In general, the impact on soils is generally positive, not necessarily always yield. It is a stable way to fix carbon, and it can be part of your soil management strategy. Now, a lot of these things, as long-term organic farmers, compost does a lot of the same types of things. It breaks down a little bit quicker than biochar, and so that's a decision you as a grower have to make if this is what you want to invest in. But in general, it will show some positives for soil health.
So, Dr. Paul, thank you for...
I've been trying. Sorry, I tried to get through it as quick as...
We got started a little late too, so...
Now we're going to talk about some soil solarization in high tunnels. And this is a SARE project with the University of Kentucky. So we have a lot of people on this one too. My research associate, Brooke Emery, and Nicole Gauthier is the pathologist at the University of Kentucky. And then her postdoc, April Lamb. And Louise Lessman is a grad student with Rachel Rudolph, who is the PD of this project at the University of Kentucky. So we know that high tunnels are high-value real estate, and they're more and more popular because we can do year-round growth, especially in the southeast. But when we start doing that year-round growth, we do tend to not rotate as well as we should because we get those really high-value crops for the summer, like tomatoes, and for the winter, like lettuce, and we don't want to give up those high returns that we get off of them. So then we might start out in a high tunnel like this, and then we start to see the diseases come in, the soil-borne diseases like southern blight and white mold and other things that start popping up in our tunnels over time when we aren't rotating like we should. we start to see some weeds. This is actually our tunnel. And if you were here this morning and heard Andre talking about transitioning their field, we kind of have the similar situation in our high tunnels, that we had a gap year in our high tunnels and the weeds went crazy. And now we have a very, very high weed population and lots of Johnson grass. So very difficult to manage now. We're gonna till the heck out of it over the winter, but weeds will come up. And then of course, insect pests and nematodes can become a problem in high tunnels as well. So the idea of this project is we want to look at soil solarization. Since this is high value real estate, could we do it bed by bed and not close up the tunnel completely? And so you could still produce in other beds and kind of solarize as you move out of a bed before you go back into it. So solarization is using passive heat and irrigating the soil underneath, making sure it's moist before you start, and then put transparent plastic tarping on the top. So it can be a good non-chemical alternative to pesticides, and it can be very effective depending on the time and temperature. So generally, soil temperatures of over 99 degrees for 2 to 4 weeks will kill a lot of those plant pathogens and plant parasitic nematodes. And similar results can be reached in only a few hours if those temperatures get as high as 113 degrees. So again, we want to look at this on a bed-by-bed basis to see if we could do it without closing up our high tunnels. So we looked at 3 different solarization times in April and July and September, in both an open and a closed tunnel, and then we did durations of either 2 or 4 weeks of solarization. And then we planted Harmony butterhead lettuce following that solarization. Then we had soil sensors in the soil to look at the temperature at 2, 4, and 6 inches. And then we looked at weed emergence, and we looked at disease suppression and insects and lettuce yield and quality. So here's what it looked like when we solarized. We had our individual beds, and we would kind of dig a trench around the edge, and then we would come in with our plastic sheeting. And if you have leftover high tunnel plastic, that's what we used. 6 mil plastic is what works well, and growers are always saving it when they take it off a tunnel, so it's a good use for it once you're done with it on top of the tunnel to use it to solarize inside the tunnel. So we dig this trench, and then we'd bury the edges of our plastic so we got a good seal under there, and then we'd solarize for 2 to 4 weeks. When we were done, we took these 1/4 meter square quadrats, and we would do biomass to look at the weeds at the beginning and then at the end after we solarized, and then every week we would look at weed emergence to see if new weeds were coming up or if we did indeed kill some of the weed seeds in the top layers of the soil. We also vacuumed up our pests once a week, so we had a reverse air blower basically that sucked them up, and then we would count the insect pests that we found inside those netting things. And here's our Harmony lettuce just before harvest. So we had 3 rows of lettuce packed in there. So again, this is going to be a little bit, oh, I think I put the wrong one in here. Okay, anyway, this is going to be a lot of slides then that are going to show you temperatures, and I tried to consolidate it, and I think I uploaded the wrong one when I did it. But this is from April in Kentucky, and so what you want to look at, what is most pertinent, is that maximum temperature here. So we were trying to get that, we want those hours over 104. And what you see here is the maximum temperature we reached in the tunnels total in April was 90 degrees at that 2-inch depth, and then in the 80s below that at 4 and 6 inches. At the 4-week solarization, it was slightly hotter, but it was still only 92. But if we're trying to get those accumulated hours above 104 or up to 113, you can see we didn't really get them in April in Lexington. Oh. But if you look in April in Tennessee, We still don't get those accumulated hours above 113 over here, but you can see that even the difference between Lexington, Kentucky and Knoxville, Tennessee, which is about a latitude change of 2 degrees, and then Atlanta would be like another 2 degrees south of Knoxville, and then Savannah would be like another 2 degrees south of Atlanta. So you can see that what the difference in temperature happens just in that 2-degree difference in latitude. So in Knoxville, we did get up to 112 at 2 inches in our open tunnel and 116 over 4 weeks of our solarization, where in the non-solarized plots that didn't have that plastic, the temperature at 2 inches was only 98 and 102. We did get some accumulated hours above 104, which is enough to kill some of our pathogens, which you'll see a little later, but we didn't get any, well, very few. At the 4-week solarization, we had 6 hours above 113. Now, if we look at the closed tunnel, you can see the difference in between the open and the closed tunnel. In Kentucky, they got to 104, basically at 2 inches. And they did start to see some of those hours, accumulation hours above 113. So they had 6 hours for the 2-week solarization and 14 hours in the 4-week solarization, and a good number of hours above 104. But you can also see, while they had 51 hours at 2 inches, they only had 16 hours at 4 inches and 5 hours at 6 inches. And that's oftentimes when we talk about solarizing, you need to be really careful after you solarize that you're not tilling up the soil from below a couple of inches, because you're not getting the heat lower down in that soil. So that's a really important thing to keep in mind as well. So now if we look at Knoxville, during that same time, we got up to 121 after 2 weeks and 126 in the solarized plots. And we do have more of those hours above 113. But again, it's in that top 2 inches, basically, that we're seeing that. Now, if we go to July, we do see greater accumulation of temperature and hours in Kentucky as well as in Tennessee. So you're getting the idea here that different times in the season, we're getting higher amounts of accumulation. Here's our open tunnel in Knoxville. So we had a good bit of accumulation above 113 in our solarized plots, but in our non-solarized plots, we didn't have any hours over that. In our closed tunnel, we started to get really hot in July here. So in Kentucky, they were above 115 for 2-week solarization and have a good number of hours that would do a good job on those soil-borne diseases. In Tennessee, by July, we were up to almost 150 as our high temperature in 2-week solarization, and we have over 100 hours for both the 2-week and the 4-week solarization, as well as at every single depth that we have there. We don't have the data for Kentucky for September yet. They're still working on that. But you can see in September, we went back down. So July was definitely where we got the most heat accumulation. We were hoping in the tunnel we would see that same effect, that we could solarize at different times of the year and still get that effect. But that's not what we're really finding. So here's the open tunnel in September. And again, what we're not seeing is that sustained heat in both the open and the closed tunnel. So unfortunately, it's looking like you would still need to close up the tunnel in order to get the effect, which is against what we wanted to do. But this is 1 year of data, so we're going to repeat it again next year and we'll see what we get. But you can see here again a good number of accumulated hours in the closed tunnel in September as well. But it's just that you'd have to close it up. So here's some of the weed counts in Kentucky. I didn't show you for Tennessee because, as I mentioned, our weeds were very plentiful. So this is in Kentucky, their non-solarized control there on the left, and what it looked like after 4 weeks of solarization on the right. So it did a good job of clearing out the vegetation for them. And here's the weeds in the open tunnel, non-solarized, and then 4-week solarization. So if we look at the dry weed biomass after solarizing, you can see here on the right, for April, we see a significant reduction in the biomass in both of the solarized, the 2-week and the 4-week, compared to the non-solarized 4-week there. And in July, same thing, we see the solarization really knocked back the weed biomass in the tunnel. So it's reducing a lot of our broadleaf weeds. We didn't have that same luck with Johnson grasses, where the rhizomes are protected and they just come back as soon as we get them out of there. And this is weed emergence from the lettuce from the time of transplanting until harvest. So in the closed tunnel, you can see there's a lot more weed emergence. In the non-solarized, this blue bar versus either of the solarized time points. In the open tunnel, it was pretty uniform, but a low amount of weeds in their tunnels overall. In Tennessee, we had, and they had this in Kentucky too, but we weeded half of our plots because the weeds just came back, and if we hadn't weeded them after solarizing, we wouldn't have had any lettuce. So that was the difference for us. But in July, again, you can see that you do get a reduction in the weed emergence in the 4-week solarization there. As far as pests go, in April, there weren't any differences in the closed tunnel versus solarizing or not solarizing, but they did find that after 4 weeks of solarization, there was a decrease in the number of pests in the tunnel. In July, same thing. While this wasn't significant, you can see a trend toward lower pests in the closed tunnel where it was solarized. As far as yield goes, we didn't really see differences in yield among treatments, whether they were solarized or not, for April and July. But diseases are one of the things that people are most interested in because they can be most problematic. Andre was also talking earlier this morning about fusarium in the soil, and that can be a really tricky one to deal with, especially for organic growers, but really for any growers. And in high tunnels, we often have limited pesticide options, no matter if you're organic or not. And there's a wide range of diseases we can get, and that lack of crop rotation really helps build up these pathogens over time. So sclerotia are these overwintering bodies. They look like little mustard seeds on top of the soil, usually right around the base of the plant. And they're great for overwintering because they're resistant to both heat and to cold. They're resistant to UV, and they're resistant to pesticides. So that's why they can just form when the conditions become right again. They start to proliferate. Southern blight produces these sclerotia. You can see them on this tomato here. But this one can affect the stem or the crown of the plants. It's going to cause rot and wilting and stunting and plant death. It has a huge host range, over 500 species, and about 30 to 50% yield loss. So significant loss in a tunnel. Rhizoctonia is another one. It infects the roots and crowns and can stunt the plant or wilt it and cause plant death. You can see it on the lettuce there at the bottom. It also has a wide host range and can cause 10 to 50% in yield losses. Fusarium is the other one. It enters the vasculature of the plant. So if you cut open that plant, you'll see this browning going right up the stem. But it'll also cause yellowing, like you can see here, and eventually plant death. So it can cause about 25 to 50% loss. So what they found in Kentucky, they took packets of the sclerotia and they planted them in their high tunnel to see if they could kill them with the solarization. We didn't do this in Knoxville because our pathologist took a job with the USDA shortly before the project started, but they did this in Kentucky, so we just hired a new pathologist, and I'm dabbling with maybe trying this, although I'm nervous to bury the sclerotia in our tunnel at all. But since we do see that extra heat bump in Knoxville, I'd be really curious what kind of results we would get for this. So what they found for southern blight was that the July solarization of either 2 or 4 weeks reduced disease by 90%. So that's really a great finding of this, especially looking at their temperatures versus our temperatures. So I'm very encouraged by this. April and September were not hot enough to knock back the southern blight. In Kentucky, the Rhizoctonia is the easiest pathogen to control. It is controlled around that 104 degree mark. So they found a 50 to 75% reduction in Rhizoctonia in April, no disease in the solarized plots whatsoever in July, and only 5 to 10% of survival in September. So, Fusarium, again, the hardest one to get, but survival was reduced by about 40 to 50% in July, though there was minimal reduction in April and September. So, the maximum soil temperatures were at least 10 to 16 degrees higher in the solarized treatments compared to the non-solarized controls. In April, really too cold to successfully solarize in an open tunnel. In July, solarization was effective in both open and closed tunnels, but we have better weed control in those closed tunnels, and the lettuce yield tends to be higher in those treatments regardless of tunnel. For September, we're still looking through the results for that, but we will repeat this study in 2025, so hopefully we'll have more results to come. And I'd be happy to answer any questions if I have a minute.
We do have time for
Okay. Tim.
Obviously, because you did research and had to do individual plots.
Right.
If you were growing in a tunnel, would you think solarizing like midsummer? Would you solarize the whole thing, or maybe do half of it? Do you think it would not be effective if you only did half?
No, I think you could do half if you wanted to. Yes, the point of this was to try to see if we could just do it bed by bed to make it easy as you are taking out, one crop before you put in something else. That doesn't seem to be like it's going to be effective, at least based off of last year. So in that case, yeah, you could just go in. A lot of people do get out of their tunnels in mid-July because it's just that hot in the tunnel, and who wants to be in there? We would walk in and come out dripping in 5 seconds. So yeah, it's not a fun place to be, but if you could close it down for 4 weeks, you would get really effective control, I would think. And that's the thing, you have a little area around where you solarize that's not quite as effective. So if you did it all in one sheet, you would have much better results overall. But we were just trying to see if we could do a little bit instead of a lot. Juan.
Good presentation. We've done in Tipton, South Georgia, research on solarization, and we were not very successful. I think, basically, long story short, compared to other parts of the country that are drier, like California and Arizona, where solarization is very effective, since we are more humid, it's very difficult sometimes to reach the temperature you need to inactivate the pathogens.
Did you do it in the field or in a tunnel?
In the field.
Yeah.
In the field. Yeah, in a high tunnel you probably get higher temperatures, but still, it's always a challenge.
It is.
You want to concentrate in the summertime.
Right. Mm-hmm.
Don't even try spring.
Right. No, that's what we thought. We thought with the added heat in a tunnel, we might be able to do it in a different time of year just to make it more palatable to growers to take it out depending on what their rotation is and when they had a little window they could squeeze it in. Shin?
Very interesting. I have a big question regarding the soil moisture content.
Uh-huh.
At the time when you actually started to do
It was 70%.
70%.
Yep. It was very moist. It was
Is that like volumetric water content or?
we had our soil sensor that told us that's what it was.
That's when you started?
Yes. So we didn't irrigate after we started. So we made sure it was good and moist, and then we took up the irrigation, put down our plastic.
What's your recommendation in terms of reaching a particular level of moisture before starting the treatment?
That's the target that we wanted, but it was highly variable. Even though we had our sensors, you could tell different parts of the tunnel. We laid our drip tape in 1 foot increments so we would get good wetting all the way across. But it still wasn't highly uniform. Rachel Rudolph has done a lot of work with solarization in Kentucky, and she's tried overhead irrigating, and she thought it was more uniform to do it this way than overhead irrigating, but I think it would depend on your system and what makes sense for you. But it was pretty painless to do it with the drip tape. We just saw that we had pockets that were more variable. That was like 2 inches deep. Yeah. Yep. All right.
Our next speaker, we're going to welcome back Dr. Tim Coulong from the University of Georgia, and he's going to give us a brief overview of water spinach.
All right, thank you. I'll have a little less data intenseness in this talk than the biochar talk, and probably I've got a little bit more time to take my time. But I'm talking about water spinach today as a potential new crop from Georgia. This is a project that's funded by the Department of Agriculture, and I'll get into kind of why and so forth here in a few minutes. As a couple people already pointed out, looks like a morning glory, Ipomea, because it is in that family. So what is water spinach? Throughout parts of Southeast Asia and China, water spinach is grown as a leafy green and often sauteed down. This is actually a picture from my student Shelby, who's working on this project, that she was cooking it up at home. And so this is right before it gets cooked down, but it's used throughout Asia. It's a staple leafy green in Asian cuisine, and it can be grown 2 different ways. Most common, hence the name water spinach, is it'll be in waterways, floating. It's hollow stemmed, floats on the surface, it roots out readily, it can be harvested, but it can also be grown in the ground and so forth. So here's the thing. So water spinach is classified as a noxious weed by USDA. And so therefore, it's not allowed to be grown in a lot of places. It's grown commercially in Texas. It's grown in Florida in a couple of nurseries. And you can see here, this is a picture from Bugwood, and this picture is from Florida of it growing. It's been naturalized. And you can see this is a map here showing the areas in the United States that's been naturalized. And so it can be grown both in soil and water, but it is an aquatic weed. When it is a weed, it's an aquatic weed. So you'll notice on this map, being in the south, it loves the heat. And so it's not frost tolerant at all. So I don't think there's risk of it becoming an aquatic weed as we get further north. But certainly, if you're a Georgia resident in the Valdosta area or something like that, it absolutely could become a weed. So why are we looking at it? Well, back a few years ago, a lot of our Asian population in Atlanta could not purchase water spinach. It was illegal to be sold. And so there's a woman who actually had a petition, I think, with 100,000 signatures, petitioning the state at least to be able to sell it legally. And so here's a cut from our Department of Agriculture. A press release here that grocery stores and restaurants can now sell water spinach, so they can import it and sell it. We still cannot grow it yet. However, after this happened, we worked with the Department of Agriculture, Juan Carlos and myself, to fund some research so that if and when it can be grown, what would be a responsible way to do it, and would we have data to give to growers for water spinach. And so that's what we've been trying to do the last couple of years. So a little bit more about the crop. It's Ipomea aquatica. So Ipomea, sweet potato, you saw the leaves look pretty similar. There's 2 main types that you can distinguish based on leaf architecture. There's one that's more of an upright leaf and one more of a flat, wider leaf. The more upright type is used more in Southeast Asia. The flat type is more apparently in southern China. And they have different colored flowers. The flat leaf type grows a little bit more aggressively and so forth, but they are, outside of that, they're fairly similar. So in terms of growing it, production can either be from cuttings or seedlings. To reduce the chance of trying to bring cuttings in from another state or elsewhere. We just bought seeds. And even though you cannot grow it legally in most states, the seed are readily available on Amazon and a variety of other sources. So that is where we got our seed. And there's no varieties. And so we just purchased, we kind of got lucky. We just purchased from 2 different suppliers, and we were able to get the 2 different leaf types. But it's not like peppers where you can order an Aristotle or something like that. It's just sold as water spinach and so forth. So this is what they look like. Come up, very much look like a morning glory type cotyledon on them. This is a Speedling 200 cell tray. This is about a 3-week-old transplant. And so what we proposed, Dr. Diaz-Perez and myself, is to grow this either in a high tunnel or a greenhouse situation so that, because it does have the potential to be a weed, can we keep it in a controlled environment? And so we did greenhouse hydroponics, and this is some of the pictures that Shelby took. What we did was we grew them in tubs because we're looking at different nutrient regimes, and we're trying to figure out what we would make a recommendation to a grower if they were to do this. And so this would be if you were growing it in a pond type hydroponic system. And you can see it puts on a heck of a root system. This is just after a few weeks in the tubs. And these plants, actually these plants are, I think, in that picture, probably about 40 days old. So they're very aggressive growers, and they love the heat. And so I'm going to show some high tunnel slides in a minute, but Dr. Weslacki was talking about July is not a good time to be in the high tunnel. But there's one thing that really, really likes it in the high tunnel in July, and it's this crop. And so one thing that we are looking at, like I said, with the different fertility regimes, is that if you look through some of the literature, it talks about this is a weed. It doesn't need a lot of fertility. But we found that it responds very positively, in fact, to fertility. And so this is a 60 part per million nitrogen here. And you see it doesn't look very good at all. This is 120 part per million nitrogen. Looks substantially better, and we saw that throughout both varieties. Thought this was kind of a dramatic picture, but I think if we're going to make a recommendation, we would suggest using programs similar to people using lettuce and stuff like that when they're growing it in a pond system to have a little bit more fertility in there. In terms of production in a high tunnel, this is one of our organic high tunnels. This was all certified organic. That's another thing. Obviously, this was not organic using mineral nutrients and hydroponics, but because it's not a crop that's grown, there aren't labeled products out there available for it. You would have to use a generally recognized as safe type product. And so growing it or for effective purposes, except for fertility, you're going to grow it organically. And so what we did was, this is a high tunnel here. We looked at different planting dates. And the idea was, if you are going to plant this in the summer, would you be better off planting it in May and harvesting it multiple times? Or over time, would that plant kind of deteriorate? Would you be better off going in and planting multiple times in the summer? And so that's what we did. We had 3 different planting dates in late May, June, and July. We had the 2 different varieties, and we did cuttings throughout the summer to compare. You can see here we hadn't planted our 3rd date yet. Grows very aggressively, like I said, loves the heat. We grew this. We had 100 pounds of nitrogen using a nature-safe 10-8, and so forth. Here's some pictures from our harvest. At peak production in July, Shelby was harvesting in about a 10 to 14 day window between picks, and you'll see when she harvested, she cut them down basically to the ground, left a few leaves sticking out, and it resprouted. But doing the math, you can't see it here, but this is 18 pounds of water spinach. We averaged about 3/4 of a pound per plant in foliage in about that 2-week window. So these things really, really put on growth very quickly and so forth. And so this is typically, these were a little bit bigger than maybe if I was going to market, but this is kind of what we were aiming for, a set of shoots like that. I will say it is an extremely heavy water user. Not surprising given the name, but we actually were up to 4 hours a day, 7 days a week with our drip. And it was still hard under those very aggressively growing plants to keep the moisture levels up. And so it uses a tremendous amount of water. So I wanted to cover a few minutes here, its potential as a weed. So the plant will flower and set viable seed. It needs to outcross, and so in the greenhouse, those plants did not set viable seed because we don't have any pollinators. In the high tunnel, when it did flower, I did collect seed that I did grow out, and it did set viable seed. So that is something to keep in mind. Freeze killed it completely in December. It thrived. It really slows down once you get into the fall, but it's not at all frost tolerant. So if you did grow it in a controlled environment and not in a waterway, you would probably be fairly well protected in terms of keeping it out of the ecosystem. It does get diseases and insect pests like anything. When I started doing hemp work about 5-6 years ago, and everyone's like, oh, hemp's this, it's a weed, it doesn't get any problem, but it gets tons of problems, right? This gets problems too. So last year we found in late 2023, Cercospora leaf spot was identified by our pathologist on the foliage there, and it was pretty significant actually. The wider leaf type was definitely more susceptible to it, but both of them did get it eventually. And armyworms. Once they got in there, they absolutely, we did spray BT products, but they could have defoliated sections of the high tunnel. The armyworms would be, I would expect to be, if this is grown, a significant pest for our growers and so forth. Also, deer. This year, Shelby was doing her study, and deer got into our high tunnel, and deer really, really liked this crop, much to my chagrin. So in conclusions, it's incredibly productive. It's not trouble-free, but I think if it is allowed to be grown with regulation, I think it'd be both effective in the greenhouse and high tunnel. If I was growing it, I'd probably do it in the high tunnel just in terms of the inputs that you would use in a greenhouse hydroponic crop, but I think both would do just fine. And with that, wrap up, take any questions. This was funded by a specialty crop block grant by the Department of Agriculture, a sustainable protected culture system for water spinach, and Dr. Diaz-Perez, who's going to talk next, is also repeating a lot of this work down in Tifton. So even hotter in the high tunnel. So with that, I don't know if I have any time. Yes, sir.
Why do you think there's no varieties? Nobody has done the selection, or?
I think, at least for what we can get, I don't think anyone's done selection. I'm sure if I went to Southeast, like Thailand and stuff, I'm sure I could get some local available varieties, but it's just because it's not allowed to be grown in the States, no one's really done any selections with it. And they're all just OP type varieties. Shin. That's up to the Department of Agriculture. I do think, realistically, if you grew it in a protected culture environment, I think the risk for it to spread is pretty minimal, if that. Well, I know now in Georgia they can purchase them. I don't know if they come from out of the country. I do know when we started doing this, that there is a group of farmers in Texas, outside of Houston, that grow a fair amount. Now, whether or not they actually export it to Florida and Georgia or not, I'm not sure. But it is grown in a few pockets.
Any other questions? All right. Thank you, Dr. Pilon.
Thank you. All right, I'm going to go run the watermelons now.
Perfect. All right, our next speaker is Dr. Juan Carlos Diaz-Perez from the University of Georgia. He is down at the Tifton campus, and he's going to talk to you about shade cloths and his work in shade cloths.
Well, thank you, Ashley, and thank you, all of you, for staying around. I'm sure you want to go out and walk in the cool conditions outdoors. Anyway, All right. I will be talking about a technology that, first of all, that is not used much in Georgia. Shade technology or shading nets are common in many parts of the world, especially in tropical countries, some subtropical countries, and some in the US, but not so much for vegetable production, and particularly not so much in Georgia. I think there is potential, and I will try to show you some data, at least to make you think about it, okay? Why shading technology might be important? Well, we've been seeing over the last 100 years or so an increase in temperature with ups and downs, and I would say a changing climate. Although I know the climate of discussing climate change might be a little bit hot, but the values in the field show increasing temperatures. That's, I think, a reality. That's in great part caused by the term greenhouse effect. Basically, gases like CO2, methane, and nitrogen oxides capture some of the infrared radiation from the sun and result in increasing temperatures. But why temperature is important? We always hear about temperature, but may not think about why it is important. Well, temperature has to do with the rate of the biochemical reactions, okay? For instance, if you have a crop, you plant some, whatever crop, how about some water spinach, okay? And the temperature increases, that's gonna make the rate of germination and growth faster. That's basically what the Van Hoff law tells us. And we can see it exemplified here with the tomato. A tomato seedling shows an increase in growth as we increase the temperature. But that increase in temperature is not like money, you know, it doesn't go forever. There is a limit on how much plants can grow. If it gets too hot, then you get a decrease in temperature, okay? That applies to humans as well. And no wonder why we're having some issues. But anyway, I won't get too far away on that. Light. I want to also tell you about why light is important. Light is energy, just like temperature. Temperature is energy. Basically, temperature is the vibration of the molecules. That's what creates the heat. Light is also vibration. If you remember from your physics, it can be as a wave or as a particle. If you are good, you like to read about this quantum physics, you can see about that, okay? So both light and temperature are energy. But why is that important? Well, you know, energy, the light from the sun is what drives life in our planet. But it's also what drives your yield in your crop. Light is important for photosynthesis, okay? The more photosynthesis you have in your crop, the more yield. So and how can we enhance that? That's something that sometimes we don't think about. Plants photosynthesize as a function or in response to environmental conditions. So you want to create the conditions that are proper for the plant to maximize photosynthesis. That's basically what it is. How do you do that? Water management, fertilization. If you could control temperature and light. If you can handle that, you're doing well. Well, of course, the pathologist might say, oh, but you have to do a good pathogen control. Well, yeah, and insect control, of course. But guess what? Pathogens and control develop as a function of these environmental factors. So everything is interconnected. Agronomists, horticulturists, for centuries have done modification of the environment to enhance plant growth. It's not recent. You can find a lot of literature on that, of how farmers have done these modifications in the history of agriculture. But why is that? Because basically, especially with techniques such as the use of plastics, what we want to do is to maximize the capture of energy. Again, energy from where? From the sun. And why energy? Because energy is photosynthesis, is sugars. That's going to be yield. The more energy your crop capture, the more yield you're going to have. How do we do that? Well, that's precisely what we're trying to understand. How can we maximize that capture of energy? One way, for instance, we modify this capture of energy by the use of mulches. If you look at this, we did some research years ago and we found that black mulch reflects 10% of the light. Silver mulch, 50% of the light. That affects the temperature in the soil and the light microenvironment around the crop. And no wonder there's some research showing that with silver mulch, you can deter some insects. I mean, there's a lot of implications on this, but keep in mind the manipulation of the environment. And that's where the shade nets come. Why are shade nets important? Or how can they be useful? Well, if you use shade nets, you can have some reduction of the heat stress. Your water management is going to improve, and we're going to see why. Basically, plants use less water. The quality of the crop increases because plants are less stressed, let's put it that way. The insects and diseases decrease. I'm going to show you some data on that. And shade nets can be combined with other protected agriculture technologies. For instance, you can use shade nets and greenhouses, shade nets and high tunnels, shade nets and some, what do you call, to protect you from water. I forgot the name in English. But why are shade nets useful? That's the question. Why? Well, basically, there are two questions that come to mind. Either light as a quantity having an effect, or light as a quality, color. We did some study trying to understand that. This study was done something like 12, 15 years ago in Tifton, trying to understand how light quantity affects bell pepper growth. We conducted the study there in Tifton, South Georgia, and we evaluated these light levels from 0% shading to 80% shading. And this is what we found, that the shading reduces, as we can see here, leaf temperature. It also reduces root zone temperature in the soil. And there's also some reduction, but less dramatic, of the air around the crop. So shading affects environmental conditions. But does that affect plant growth? Yes. How does it work? Well, you can see here, it makes the plants to have thinner stems. The more the shading, the thinner the stem in the plants. As we increase the shading, the total plant leaf area increases. Why is that? Plants have antennas to capture light. So if the light level decreases, plants need more antennas. It's like when you're looking for the signal for your cell phone, huh? Okay? You need antennas. So plants do that. They adapt to the level of light. As we increase the shading, the number of leaves per plant decrease. That's funny, eh? We have more leaf area, but less leaves. But the size of these leaves increases. That's why 100 years ago, when people started using shade net technology in South Georgia and also in Florida, they used to do a lot of shade tobacco because shading increases leaf size, which is what you want for shade tobacco. Okay, look at the plants in this case. You can see the effect just visually going from 0% to 80%. You can see the gradual increase in plant size, which is precisely the data I showed you. What happens physiologically? Well, plants tend to have stomatal conductance is basically how open the stomata are. And we can see that the stomata open at medium level of shading. That is telling us when the stomata open in a plant, it usually means the plants are happy. Okay, they have sufficient water conditions in the soil to allow them to open the stomata. When the stomata close, it's usually the plant trying to protect its water, so they close the stomata to prevent dehydration. So this opening of the stomata at medium range shade level, telling us that the plants are doing better at the medium range, okay? It's also telling us that the plants may be using less water, because there is, at this level, temperatures are lower compared to zero, open air, open field. So we are modifying the evaporative demand of the plant with the shade technology. Photosynthesis goes down. This is interesting. Look at this. Up to about 30% compared to 0%, photosynthesis is about the same. But then at higher levels of shading, photosynthesis closes. Of course, plants need light for photosynthesis. At higher levels of shading, there's not enough light, plants don't make photosynthesis. Consequently, what do you think is going to happen with the yield? Tell me. Yield is going to go up or down? Down. Okay, we're going to see that now. Fruit number decreases with shade level. You got that? Less number of fruit. How about the yields? Let's see if gentleman was right or wrong. Medium range, in this case of bell pepper, highest yield with 30% shading. When the plants were happy, remember I told you the stomata were open? Well, yield is the highest. But then as we increase the shading, katapoom, the yield declines. So do we have this area between 30–40% and no shading that can be fine-tuned to increase yield without impacting photosynthesis, allowing the plant to have sufficient light to function. One problem with bell pepper is sun scald. Well, we found that 30% shading or more reduces the sun scald, okay? Which is a big problem in South Georgia. So it does help to reduce that. And also increases fruit size. If for some reason you're interested in having larger fruit for marketing purposes, fruit size increases with shading. And also, very interestingly, we found that shading reduced the incidence of Phytophthora capsicum, a root disease in bell peppers. We don't know why, but this was an interesting finding. And it's consistent. We've been finding also a reduction in Sclerosia rosea, southern blight, with shading. Even 30% shading reduces southern blight. So apparently there's some connection between heat stress and these two diseases. So we got some amelioration of heat stress with shading in the case of bell pepper. And we found that about 30% is what we consider acceptable or reasonable for bell pepper. I need to make a clarification here. We are in an organic session. And you might wonder, why in the world this guy is presenting data in conventional production? Well, I have to recognize and maybe propose a hypothesis that this type of responses work across conventional and organic, okay? Because we're talking about light manipulation. But let me now show you some more data. In the previous, we showed you data on light quantity, shading. In this case, I'm going to show you data of using shade nets of different color, which is changing the color environment around the plants. There were some data associated to our study or related to as a connected literature showing that, like in this study, they found that the yellow net produced the most compared to black or even red and pearl. So basically, the color of the net affected yield production in pepper. These people. Falik, I think, is from East Europe, if I am correctly. We did a study evaluating those colors, red, pearl, or silver, kind of, white, and black. That's what we did in bell pepper, with a shading value of about 30 to 40%. This is what we found. The plant height and stem diameter were very similar in all the shade nets, in all the colors, no difference. And the plants that were not shaded were shorter and had thinner stems. You can see that in this, these are unshaded compared to that, shaded. So there was more plant growth with the shading, but regardless of the color. How about the yield? Well, yield was the same. All the shaded plants, regardless of color, had higher yield in terms of fruit number and fruit yield. Also in terms of fruit size. So plants that were unshaded did the worst in terms of fruit yield. So that's what we got. So shading did benefit bell pepper plant yield, but the color of the net makes no difference. Is that important? Well, I think it is. At least based on this data, there's no need for growers to spend more money in a net that is colored compared to using a simple black net. I'm going to show you data on habanero. We did some studies on habaneros. Have you tried habaneros? They are hot. They are hot. The sauce is just wonderful. Wonderful. Shade level. Look at the response. As we increase the shade level, we reduce the fruit numbers and we reduce the fruit yield. There is an area about between 0% shading, that means unshaded, and 40% shading in which the yield is not much affected, but then above 40% shading, your yield decreases. But there's no benefit in fruit yield. I want you to make emphasis on that because basically it's telling us compared to bell peppers, this type of peppers did not benefit from shading. In a way, you can say in various aspects, smaller fruit peppers tend to be more resistant to a lot of things compared to bell peppers. So, not much benefit. I think Sunscald was reduced with the shading. That's a good thing. And there was increase in fruit size. There was increase in fruit size, but not so much benefit in terms of yield. So not so excited. So I guess the main benefit is in fruit size and some reduction in Sunscald. So two peppers responded differently. So far, it's telling us that we have to be careful about taking the result from one crop to another. It may not be the same. I'm also would like to share some data on a project in which we work with Dr. Sheen from Florida, from the University of Florida, Dr. Coulon, and we did this study a few years ago on using shade nets in combination with high tunnel, with the idea of cooling the high tunnel to grow tomatoes. And guess what? This is inorganic tomatoes. So at least I'm presenting some organic data. Okay? In conventional tomato production, we have probably a good time to grow tomatoes from March to June, and August through October, more or less. In the summer, July, part of August, but yeah, basically July, it's too hot, at least in South Georgia. Very challenging. So, one of the ideas in this proposal was to test if we could, by manipulating light, grow high tunnels in the summer. It's a very, I would say, ambitious goal. But that's what we tried. And the results I'm going to show you is not what you typically see in a textbook. Because in a textbook, everything looks great, everything looks like science is perfect, but science is not like that. But oftentimes we learn by mistakes. And I think if we pay attention to the mistakes or the things that we were not expecting, we also can learn a lot. In this study, we use a black net, a silver net on top of a high tunnel, and a hot tunnel that is unshaded. And we also use black mulch and silver mulch as a plastic mulch. First, look at this. Inside the tunnel, it made a difference whether you grow the tomatoes on white mulch versus black mulch. Black mulch was too hot, as you would expect, in the summer, planted in the fall. What happened? Well, it reduced plant growth, regardless of whether the high tunnel was shaded or unshaded. And the plants on white mulch grew faster than the plants on black mulch. Also, there was more plant mortality with black mulch. But visually is clear. Having a net made an impact. You can see the capture of energy without net and with net. Some values, air temperature with black net was the lowest, and without net was the highest. And the roots on temperature follow a similar pattern. With nets, roots on temperature was lower compared to unshaded. So what is this telling us? That definitely the nets reduce the heat load that's coming onto the plants, and that reduces air temperature and soil temperature. How did that affect the yield? Pay attention to that. What is the highest yield? In the unshaded. Is that what we want? We want the lowest yield using shading nets? I was disappointed when I see that. But you know, when you see this type of data, negative results, you do something and you end up having lower yield. You don't just stop and start crying and say, forget it. No, let's try to dig and understand what happened. She's telling me 5 minutes. That's good. No, I'm not picking on you. I'm just gonna tell you it's okay. Okay, yield decrease. Look at this. Light intensity. We know that light is necessary, right? Okay, look at the light level. This is in the open field. It's about 2,000 micromoles. That's basically normal light. This column is the light unshaded, the high tunnel with nothing on top, just the film covering in the high tunnel. Light was reduced by about 30%. Why? Why light was reduced so much? It's something you have to keep in mind as a grower. Just the film on top of the high tunnel reduces your light. And depending on the film, can be 20%, 15%, 30%. Guess what? This is probably because we didn't clean the top of the tunnel, so we end up reducing light by 30%. Our data in bell peppers told us that a 30% reduction in light is as much as you can go to have the optimal yield. So in terms of light, this is probably as low as you want to go. I mean that. About 30% shade level, or about 1,400 micromoles per meter squared per second. But when we use the nets, we lower to about 60% shade level. That was too much. 60% shade level decreased the yield in bell pepper by about 30 to 40%. Oh. Which is what we saw here. The yield here compared to that is about 30% lower. Why? Because we were similarly to bell peppers. We were in the 60% shade level. That is too high compared to normal light. So basically, we are reducing the light to very low levels with the shade nets. So, what do we have to do? Shade nets help the plants to get established because they reduce the heat. That was important. But the yield was decreased. And why the yield decreased? Because we never removed the shade nets from the high tunnel. So what do we have to do? Well, very likely, after the heat is gone away, maybe in October, remove the shade net from the high tunnel, and instead of being with a shading level of about, in this case, 60%, move it back to about 30%. That's going to allow your plants to have sufficient light, and likely, as a hypothesis, because we never tested that, your yields are going to increase. That's what we learned. So, again, the advantages of the shade nets, if you want to consider them, especially I think this might be an option for small growers who have a niche market. This can be an option because you get all these diseases. Reduction of heat stress, your quality of the fruit increases, you may have less problems with diseases, and you can improve water management because you're going to use less water in your plants. But everything is not perfect. What are some of the issues or disadvantages? Well, you have to watch for reductions in photosynthesis, light. Be sure your light levels are not too low. You have to have good ventilation, otherwise high humidity may end up increasing your risk of diseases. There can be a high initial cost, of course, and nets may need to be removed if you are in a high hurricane risk area. But other than that, that's where we are. Thank you so much. Am I on time?
Absolutely, yeah.
Good. Yes.
Me? Yes. For example, I've seen the white plastics that they use sometimes on high tunnels, at least where we come from, that is not a completely clear plastic. And it's white plastic and they have like a 50% light reduction. Do you think that is the same as using a 50% shade cloth, for example? The same type of reduction? Or one being a white plastic and the other being black shade cloth, for example, the properties are different inside of the structure?
I'm not sure exactly which plastic you're talking about. I've seen some white plastics that have some incorporation of compounds that reduce the ultraviolet light, or some others that reduce the infrared. The ones that reduce the infrared have a stronger cooling effect, at least theoretically, compared to the simple black shade net. It's a technology that is being used in greenhouses And some people have been pushing it towards high tunnels because they have a stronger cooling effect. They remove some of the infrared radiation. Yes, potentially it could be more effective than shade net, but I don't know the one you're talking about.
Yeah, but they can be different.
Yeah, so yes, the short answer is it could be. Mhm. Yes, Dr. Pica. Thank you.
Did you do any work to kind of quantify what the critical root zone temperature might be that you get inhibition of pepper or maybe tomato growth? So above a certain temperature, when the temperature of the root zone gets too high, you get reduction in plant growth and development. Any comments on that? You know, what that temperature might be in terms of the root zone that you might get inhibition of growth and development of peppers or tomatoes?
Not in this study, Dr. Pica, but we've done that work. We have done work in tomatoes, peppers, eggplants, watermelon, and we have developed the response curve. Basically, in the case of bell peppers, the seasonal mean temperature at 10 centimeters deep, around 27 Celsius, is what maximizes plant yield. 27, something like that. In tomatoes, it's about 26 and a half. And corn, maybe more like 28. So yes, there are some differences. And we've done the work, Dr. Pica. Mm-hmm.
Any other questions? All right. Oh,
Do you know what the age of your plastic was?
the age of the
Yes, when we started the project, it was about 1 year old. It was relatively new. They just put it. And the study lasted 2 or 3 years, so yes. As the time goes, you know, plastics start to deteriorate, they get stained, so your light penetration ability is gonna decrease as the plastics get older. So that's a good question. As growers, we have to measure light. That's one of the things that is a challenge right now. Growers have to measure environmental, especially as you growing in high tunnels and greenhouses, you have to measure your temperature, your light. You know, the more elements you measure, the better. Yes, so it's important. We need to understand that plants eat light. If you don't have enough light, you can put a lot of the other stuff, fertilizer, water, whatever. Plants are not gonna produce so well. Good question. Thank you. Yes, Shin.
Also wondering if you have done any research involving shade cloth of different colors besides the one that you have been using, like black or reflective or light.
Not other colors, Shin. No, I have not tried other ones.
Do you think the color may show some impact or not at all?
No, that's a valid question. Indeed, before we, no, that's a very good question. Before we did the study, I was enthusiastic about some research in Israel. You know, Josefa and her group in Israel, in the Volcanic Institute, have done significant amount of work on shade net color. And they were promoting the use of a red net as a increasing or enhancing photosynthesis, plant growth, and yield. And so we use that type of, the red I use is from that group. But what we found is it didn't show any benefit for us. Let me give you some background, if I may. Okay, a few years ago, when we started to do the plasticulture work in Tifton, about 25 years ago, there was a mulch, plastic mulch, that was very famous. It was a red mulch. And again, there was a lot of research on the use of red mulch because supposedly increased quality, and I remember some work in strawberries, increasing sweetness, and in tomatoes, yield, and good work. And again, I was very enthusiastic. So that's how I started to do my work on plastic mulching using different colors. Well, guess what? I guess Tifton is a party pooper. We didn't see anything in Tifton, and we tried it different years. And what our data showed is that the differences in response in Tifton, I don't want to I don't know anything else, was due of the mulch acting in the effect of the mulch on soil temperature was causing the response, not the effect of the mulch on the aerial light environment. And how can I say that? Because we use the mulch in the spring and in the fall. Using it in the fall, temperatures in the soil were higher, plants produced much less. Using it in the spring, plants did really well. So what changed the most? Light environment did not change much. Soil temperature changed a lot. That's why I say it's probably more related to soil temperature than to light environment. But again, you can see a lot of studies in the literature saying it's the effect of the light environment, and that's fair. I'm just telling you what we found in Tifton. There you go. So basically the red is like a black. So a lot of growers in South Georgia ask me, hey, do you think I should buy the red mulch? I say, well, if you want to pay twice as much and get the effect of the black, go ahead.
Do you think same could apply to the shade cloth?
Our data suggests that. But there's so much data showing an effect of the reflected light that I don't know. But at least for us, we didn't see an effect. That's the only thing I can say. Thank you.
Dr. Diaz-Perez, thank you so much.
No, thank you.
This is a
Ashley.
nice
Good
little
to meet
mug
you.
for you.